Howdy, this is Browdy. So this is the second video of Unit 3. Oh, that's not good. The second video of Unit 3, and we are following up the conversation last time about lipids and polarity, and this time we're jumping into it with the structure of the cell membrane. And so we'll see here, I'm going to zoom in a little bit actually. We are talking about cell membranes and transport specifically. And so if you haven't already gotten them, please make sure you download the notes. And this set of notes we're actually going to use for the next the next video or two. So this might be a 1 to 2 to 3 part set of notes, just because there wasn't really a good way to differentiate between all of those sections, so I kind of made it all one big thing. So we're going to be using this set of notes for a little while, and I think that'll be fun. So last time we talked about the chemistry of water. We talked about lipids, so saturated and unsaturated fats. Then we brought that all together, talked about phospholipids, so how it's a molecule that has a polar head and a nonpolar tail, and how those arrange into the phospholipid bilayer. So this time we're going to start by talking about the structure of the cell membrane. And so the cell membrane is described with what we call the fluid mosaic model. And so the fluid mosaic model, what that does is it describes the liquid nature of the cell membrane and how parts move around easily within it. So fluid refers to constantly in motion, right? Or the flow of something, a stream flows, it's in motion. Even those of you who know anything about me outside of school, right? I deal a lot with drums, and I'm a drummer, and I teach drumline, and we talk often about fluid motions when you're playing drums, right? So it's not very robotic like this. You want to play with a fluid approach, so constantly in motion. Mosaic, now I'm not the best person to vouch for the arts here, or at least art in terms of like art class and things like that, but mosaic, the way I think of it at least, refers to a bunch of smaller things kind of coming together to make a bigger picture. And so in this case, it refers to the fact that there's a bunch of different things kind of scattered throughout the cell membrane that contribute to the membrane itself. And so for those of you who have been in class, you have done a bubble lab of sorts. And for those of you who haven't, what it is we had soapy water and we had kind of a bubble wand or a square bubble, and there were a few things that we observed. You could bend it, right? You could poke things through it, and all of these relate to the cell membrane. You could also put a rubber band on it and pop a hole in the rubber band, and then still move that hole throughout the bubble. And so that is again referring to kind of the liquid nature of the cell membrane. I have at times described the cell membrane as being kind of like the balloon in a rubber balloon, or not a rubber balloon, a water balloon, which works in terms of the balloon is what keeps a water balloon from just being a puddle of water. So the cell membrane is similar in that sense. But it's also much less rigid, much less strong in actuality. So the cell membrane is much more like a bubble where it can flow and move around. And so the fluid mosaic model describes that. The things that are in it move around within it. So the cell membrane has two primary functions. The first is to make a barrier between the inside and outside of the cell. So like I said, the water balloon example works very well here. It's the difference between a water balloon and a puddle of water. So the cell membrane is very similar in that sense. The second one is to control the concentration of molecules inside the cell. And so here we can see that here's a cell that doesn't have a lot in it, and then here's all the stuff outside it, and the cell membrane is keeping those things out. And specifically, we can see the cell membrane here with all the stuff outside of it, and then a few things embedded in the cell membrane. So let's talk about what the heck is in the membrane. There's four main parts that we're going to talk about as far as what is in the cell membrane. The first are phospholipids, which we've talked about already. So these are the things that have a polar head and a nonpolar tail. They arrange themselves so the polar hydrophilic portion is on the top and bottom facing water. And then the inner portion of it are the nonpolar hydrophobic tails, and that's what keeps things from passing through. It's those are phospholipids. Second are protein channels. And so these help molecules pass through the membrane. And I like to think of these, and maybe this analogy is a little off at this point, but I think of these as the tubes from Mario. So if you ever played any of the Mario games, especially the original one, you know, there are those green tubes or pipes or tunnels, whatever you want to call them, and you go through and they go broo, broo, broo. I kind of think of the protein channels like that, right? Because you're playing Mario, you're skipping along, and then you come to a tunnel, you jump through, and it takes you down, and now suddenly the world that you end up in, the thing that was the ground before is now the ceiling, and you're down here. And that's kind of what the protein channel is it's a tube that takes you from one side of the membrane to the other. It's a tunnel, or it's a channel that takes things through, and we'll talk more about those as we go. There's cholesterol, which is a molecule that's kind of spread out in the nonpolar section, and it basically thickens up and strengthens the membrane, so it's a little bit tougher, right? So it is still fluid, but it needs to be somewhat protective, and so cholesterol serves that purpose. And then lastly, there are glycoproteins. And these identify the cell to other cells. So I like to think of these as kind of like flags or markers or almost like an ID that the cell uses to say, hi, I'm me. So let's look at these. So here's the phospholipids, right? These things, it almost looks like a bundle of Q-tips. But here's all the polar hydrophilic heads. Same thing down here. And then here's the non-polar hydrophobic tails, all facing each other. Now here's the protein channel, which is the thing that I said is like the Mario, excuse me, the Mario tube. So you could hop in here and then go through and get transported to the other side. So here's a protein channel as well. You might hear these called transport proteins. The third thing we said was the cholesterol, which is this sort of pentagon looking molecule here. And that's kind of embedded throughout to make the cell membrane thicker. And then lastly, we have these glycoproteins, which stick out as like identifiers, and they're ways for the cell to be identified by other cells. Now here, I unfortunately do not have a way to play this video for you. I've been trying to figure out how, and unfortunately just with the way that I record these videos, I can't go to a different screen without it stopping the recording. So unfortunately, I cannot play this right now. If it's something you would like to play, I might be able to link this in the video page or in the discussion page. So it is something I would love for you to be able to see, because it's a great video, but unfortunately I cannot play it right now. So I'm going to keep going just for the sake of consistency. So the cell membrane, we're moving on now. So that was kind of the structure of the cell membrane itself, but now we're going to talk about some of the characteristics of it. And so as far as the notes are concerned, we're actually only going to do this first page. But the first thing, and you've heard me say this before, is that the cell membrane is selectively permeable. Permeable referring to being able to go into or out of the cell. So being able to permeate something means to go through it. And so it's selectively permeable, meaning that some molecules can cross the membrane, but not all of them. So certain things can cross, certain things cannot. And so there's four main ways that molecules can cross the cell membrane. The first is called diffusion. So this is where small, nonpolar, uncharged molecules can slip right through the phospholipid bilayer. So the phospholipid bilayer here, again, polar heads, nonpolar tails. And so you have to be small enough to fit through this gap, nonpolar to be able to get through the nonpolar layer, and then uncharged, and then you can just kind of slip through. But not a lot of molecules are like that. So that's diffusion. That's the first method. The second is osmosis. So this is specifically looking at water, and it's where water moves through the membrane through very small water-specific protein channels, and we call these aquaporins. So aqua referring to water, and porin, kind of like pore, right? You have pores on your face or on your skin, and so you can think of pores as holes. So I think of it as water holes. So water moves through these aquaporins to get from one side of the membrane to the other. You have facilitated diffusion, which is basically what osmosis is, but that's water specific. And facilitated diffusion in general is where molecules move through protein channels to go from high to low concentration. So here you see that there's a high concentration of these molecules here on this side and a low concentration of these molecules on this side, and so they move from high to low concentration. And so this is considered passive transport, and really, referring to the notes over here, diffusion, osmosis, and facilitated diffusion are all what we would consider passive transport, meaning that it takes no energy, or they go from high to low. The last one, and actually the last thing we're talking about in this video lecture, is active transport. And so we'll talk more about the specific types of active transport, but that's where molecules move through protein channels from low to high concentration. And so this is the opposite of what you might think, right? So if there's fewer things here and more over here, normally those would go this way. But active transport actually sends them against what we call the concentration gradient. And that requires energy to do that. I like to think of this as swimming upstream. So this is active transport. So there's two kinds. There's active and passive transport. And again, passive means no energy. Active requires energy. And so that's really where we're stopping for today. But again, think of active and passive as the two kind of categories of transport. Passive, think of it as kind of going with the flow or going downstream. Or for example, if I were to take a bowling ball and stand at the top of a hill, if I just put the bowling ball on the ground, it would roll down the hill. It would go from high to low, and it wouldn't take any energy. It would just happen. But if I were on the bottom of a hill with the bowling ball and I wanted to roll it to the top, to go from low to high, that's going to take a lot of energy. So that's active versus passive. And that's where we're going to pick up next time. And I'm looking forward to seeing you in class, and I'll talk to you then.